we discussed about G protein and their mechanism of action. The G protein is asked. G protein is a protein which contains GTP, which is GTP bound protein, and which participate in signal transduction process of various membrane bound hormones. Hormones which act through membrane bound receptors. Among them, few use GTP as NAS signal transducers. See, in every sentence I have used different terminologies so that you can correlate about the different terminologies, because the question may be asked in any of these terms. The things are same, only the names are different, right? So that you can understand. Now, this whole process, sometimes it is asked in the examination process. It comes as a long question. It comes as a long question stating classify hormones and explain or discuss one of the second messengers, or explain the mechanism of G protein in hormone transduction pathway, or they can pick up a specific pathway, say for example insulin pathway, for example glucagon or epinephrine pathway, for example beta adrenergic receptor pathway. So you should be very much aware about which hormone acts through which transducer and which pathway they have asked. Correct? Epinephrine, norepinephrine, glucagon, they all act through G protein and cyclic AMP as second messenger. G protein is not a second messenger. It is a bridge between second messenger and outsider hormone. Correct? Fine. So the G protein has three subunits, alpha and beta and gamma. I repeat, alpha and beta and gamma. Why? Why it is so differently? Because beta gamma remains together and alpha remains separate. Alpha is anchored to the cell membrane through its own anchoring protein, rather molecule, not protein, anchoring molecule, and beta gamma, the beta subunit is anchored to the membrane, and gamma is holding as if it is riding on the shoulder of beta, as if it is riding on the shoulder of beta. The gamma has two extended arms, two alpha helices, through which it binds with the beta. Suppose this is a beta, so it will hug like beta like this. Okay? And beta is bound to the membrane. Gamma is not bound, but gamma is closely associated with the beta subunit. Now, the interface where alpha and beta subunit interacts, there lies a small region which is responsible for binding of GTP and its hydrolysis. As long as it is GTP bound, it cannot reassociate. As long as it is associated, GTP cannot bind. See this one. This is GTP bound altogether. Then some changes occur in this green color. What is that green color? Receptor. What is the name of the receptor? A specific name chilo. 7-TM receptor. 7-TM receptor. See, very clearly, this is very important chapter. No one will explain you after this class. Switch it on. So 7-TM receptor. The red dot is the hormone, green bar is the 7-TM receptor, and these are G proteins subunits. When they bind, see, they dissociate. See, GTP binds and then dissociates. Why? Because the GTP binding site occurs at the interface of beta and alpha. Means like this, if they are separated, I can stand in between. If they are not separated, I cannot move in. So me, if you consider as GTP and these as beta and gamma closely associated, then I cannot move in. Fine. The moment they go, then GTP is hydrolyzed. GTP is hydrolyzed, and that energy helps in triggering this dot, that is activation of adenylate cyclase. So if I ask you which subunit of the G protein is the effector part? Alpha. That is why there are different types of G protein based on different types of alpha subunit. Some alpha are termed as alpha S. S stands for stimulatory, I stands for inhibitory, T stands for transducing. These three you remember. There are AQ also there, Q is also there. Okay, at least three you remember. First one is stimulatory, second one is inhibitory, third one is for vision, different actions. Now, this cyclic MP thus produced maintains an equilibrium of the cyclic AMP concentration inside the cell. There is the enzyme which cleaves cyclic AMP and produce AMP. The name of the enzyme is phosphodiesterase. So, a balance between phosphodiesterase and adenylate cyclase is crucial to determine the concentration of cyclic AMP inside the cell. If cyclic AMP concentration is more, then remember outside the cell glucose concentration is less. If cyclic AMP concentration is less, remember outside the cell glucose concentration is high. That means glucagon is associated with high cyclic AMP. Epinephrine is associated with high cyclic AMP. Norepinephrine is associated with high cyclic AMP. The opposite is insulin. But as a matter of fact, insulin has a different pathway. Insulin does not follow cyclic AMP pathway. Alright? But insulin helps to stimulate phosphodiesterase, which will radically reduce the concentration of cyclic AMP inside the cell. It will not inhibit cyclic AMP producing enzyme. It will stimulate cyclic AMP destroying enzyme. In that way, it will reduce the cyclic AMP concentration. Fine. Now, the cyclic AMP is produced. Now, this thing again should reunite. So, what you need to do? GTP is hydrolyzed and GTP is removed and this space is open. Now, they can reassociation of subunits. So, this is the flow chart of activities taking place through a G protein. Now, we come to second messenger, those who present a signal, synthesize release from storage, intracellular ligand, act as intracellular ligands, commonly use a G protein coupled receptor, signaling made or released by effector proteins. Ligand for GPCR is first messenger. Second messenger's features of second messengers. What features you should look for to call a molecule second messenger? Are they in low amount in resting state? If yes, they are second messengers. Are they regulated? Are their synthesis is regulated by higher molecules, some other enzymes? Ultimately, whether they are under parental regulation or not. Fine. Whether they are at home or at hostel. At home, homely. At hostel, hostile. So, at hostel, they are hostile. So, they are under the parental regulation. The regulated destruction. That means what? Synthesis and breakdown. Whether these processes both are present or not. That means it is only being synthesized and cannot be regulated. It's not second messenger. Then what will happen? A cell will remain in a particular metabolic state forever. Your eyes will shed tears even in the happy newses. When you are crying, when you are sad, it shed tears. When you are happy, then also it is shedding tears. Because it is uncontrolled, hostile. Act through other proteins. They do not perform any action. They trigger the action of other molecules. They are not the king. They are kingmakers. They are not the king. They are kingmakers. They do not do anything. Right? Now comes the second long question from this chapter. Enumerate different types of second messengers. Explain any one of them, or any mechanism of action of any one of them. Any one you can take. Fine? Each one we will learn separately. First one is cyclic AMP, second is cyclic GMP, third one is calcium ion, fourth one lipid derivatives, IP3 or PIP2. IP3 or PIP2. Then DAG means diacyl glycerol, and many others we will see. Starting with cyclic AMP signaling pathway. লিখবো? লেখা হয়নি? হয়েছে? চেঞ্জ করি? Cyclic AMP signaling pathway. This is a cell membrane, receptor, G protein, effector, ATP, cyclic AMP. What is the name of the enzyme? E. What is the name of this enzyme? E. Adenylyl cyclase. Adenylyl cyclase. Be with me. You are so less in number. Just be with me. It is just like a private tuition. No, you are getting a private tuition at nominal cost. Just imagine each of you has to pay 1,000 rupees. Now you are paying only 50 rupees. So take the advantage. Huh? pKa, protein kinase A. Protein kinase A, B, C, there are different types of protein kinase. So cyclic AMP stimulates protein kinase A. Protein kinase A converts. Protein kinase means phosphorylation. So it stimulates phosphorylation of something. Some substrate is phosphorylated to the phosphorylated product. The substrate is phosphorylated to substrate P. PDE, phosphodiesterase. The same enzyme which act on cyclic AMP are also destined to dephosphorylate the other phosphorylated substances, because it's a balance. Always there is a balance, production and degradation, right? It's a dynamic process. Now if one of these plus or negative sign means one of these enzyme is activated more, that means that product will be more. If this enzyme is this step is preferred, then this product consumption will increase. If this step is preferred, then this product will increase. Clear? Now, this is an overall chart. দেখে রাখো, পরে আমি আসছি। নাম্বার 1, এখানে কি যে নাইট্রিক অক্সাইড, এটা এখানে দেওয়া আছে 2 নম্বরে। গ্লুকাগন 1, G protein, cyclic AMP, protein kinase A, protein substrates. Now this protein substrate, let me ask you, give one example of protein substrate that are phosphorylated in the effect of glucagon from your previous knowledge. Recall the glycogen metabolism, regulation of glycogen metabolism. Glucagon promotes which pathway, synthesis or breakdown? Glucagon কখন secrete হয়? যখন ব্লাডে গ্লুকোজ বেশি থাকে না কম থাকে? হ্যাঁ? দাঁড়া তুই দাঁড়া। গ্লুকাগন কখন secrete হয়? ব্লাডে যখন গ্লুকোজ বেশি থাকে না কম থাকে? হ্যাঁ? কম থাকে। শিওর? আচ্ছা। তাহলে কোন পাথওয়েটা যখন হবে তখন হবে? গ্লাইকোজেন ব্রেকডাউন না গ্লাইকোজেন সিনথেসিস? তুই না খেয়ে আছিস, তোর মা খাবার টেনে আমাকে দিবি, তোকে দিল না, কি করা উচিত? আমাকে দেবে, তোকে দেবে? তোকে দেবে, হ্যাঁ না? তাহলে ব্রেকডাউন হওয়া উচিত। তাহলে গ্লুকাগন ব্রেকডাউনকে সাপোর্ট করে। এবার যদি মনে করে দেখো ব্রেকডাউনে এনজাইম নাম কি? ফসফোরিলেজ, তাই না? সিনথেস এনজাইম নাম কি? গ্লাইকোজেন সিনথেস। মনে করে দেখো গ্লাইকোজেন সিনথেস এনজাইমটা যদি ফসফোরিলেটেড হয় তাহলে সেটা ডিঅ্যাক্টিভেটেড হয়ে যায়। সো গ্লুকাগন ডিঅ্যাক্টিভেটস synthesis. Glucagon promotes phosphorylation of phosphorylase kinase. Phosphorylase kinase promotes phosphorylation of phosphorylase. Phosphorylase promotes glycogen breakdown. Clear? মনে রাখবে insulin causes usually not in all cases but usually insulin causes generalized dephosphorylation inside the cell. When we talk about hormone, it is inside the cell. When we talk about enzyme, it could be inside as well as outside the cell. Hormone means always inside the cell, intracellular, be it through receptor, be it through intercellular receptor, doesn't matter. But it always must act within the cell. Outside the cell, hormone has no action. Unlike enzymes. Now, so protein substrate is glycogen synthase, one example, which is deactivated by phosphorylation. So one pathway, transduction pathway is complete. Come to the second one. This is nitric oxide. It acts to cyclic GMP. What is nitric oxide? Vasodilator. Is it solid, liquid or gas? Solid, liquid or gas? জোরে বলো। Gas. Have you ever smelled nitric oxide? So nitric oxide being gas, does it require cell surface receptor? What do you think? God and ghost, do they require doors to come inside? God, G for God, G for ghost, and G for gas. They don't need doors. They can easily enter anywhere. Fine. That is why ammonia is converted to urea. Otherwise, there will be toxicity in the CNS because ammonia is gas. It can be God, it can be ghost. Both. When you start the letter with G, do you know what you are going to end with? Whether you are going to write God or you are going to write ghost, you don't know. You start with G. Right? Fine. So, cyclic AMP is activated through intercellular receptor and then PKG, protein kinase. G means what? G protein associated protein kinase. The protein kinase which will act on G proteins. One is guanylated proteins. So protein substance is activated which brings about vasodilation. Number 3, come this T cell activation. Say suppose a cell surface receptor receives a ligand and then it activates through G protein which produces calcium ion. From where? Bolo? Endoplasmic reticulum, very good. Any other way to bring in calcium? Calcium channels, they can open calcium channels, but these will produce calcium inside the cell. I am talking about within the membrane. The calcium ion must be within the membrane, otherwise it cannot travel further. The calmodulin, this is very difficult to understand. The calmodulin, it's in intracellular compartment, but it is closely associated with membrane when the action is there to happen. Means, otherwise throughout the year, the hooligans, the gundas, they don't create problem. Once the election approaches, until election approaches. The moment election approaches, they go and get attached with some or other membrane. Membrane means some party, political parties. So they come associated with some membranes, and then their effect come across. Otherwise, calmodulin is free. The moment they get associated with the membrane, the calcium produced in the membrane is directly associated with calmodulin, and they bring about certain changes to do the action. Throughout the year, the gundas will be very timid because police will beat them. The moment election approaches, police will become selective. This is our nation. They will become selective. Which one to catch, which one not to catch. So you be selective during election. So calcium, calmodulin, and then multifunctional kinases are activated and protein substrates are produced. The same thing will happen in case of diacylglycerol. The activated G protein might produce diacylglycerol from IP3, inositol triphosphate. Inositol triphosphate. From where? Phosphatidyl inositol. From where? Cell membrane. In the cell membrane there are phospholipids. The first one is usually, first fatty acid is usually saturated. The second, C2, is usually unsaturated. And third one is a phosphorylated product. What is that? It may be serine, so we call phosphatidyl serine. It may be choline, we call phosphatidyl choline. It may be something else. Only phosphate, phosphatidic acid. And it may be IP3, then phosphatidyl inositol. Okay? Phosphatidyl inositol triphosphate. So this is a phospholipid. And this phospholipid IP3 is released. If you remove the third fatty acid, or third phosphate group, then we are left with what? A DAG, diacylglycerol. So DAG and PIP2, they are produced simultaneously. Correct? So there is a small piece of bread and also a small piece of sausage. And I butcher it, break it, cut it with a knife. The two parts are separated. One is small part is IP3. The big part is DAG. DAG has separate pathway of action. IP3 has separate pathway of action. So one movement, two different actions can be generated. From same product, from same molecule. So they stimulate PKC. They phosphorylate a certain protein substance and bring about changes. Calmodulin, phospholipases are activated. See, something goes wrong in the membrane which let the calcium ion enter. Which let the calcium ion enter. Now this calcium ion through this pathway will stimulate what? Phospholipases. In order to produce DAG from phosphatidyl inositol triphosphate, which phospholipase we require to stimulate? Try to recall different phospholipases. First one, if we cut at C1, it is A1. If other side of the C1, then it is A2, then B, then C, then D. With phosphate, without phosphate, D, this side, that side. So it is phospholipase C. আচ্ছা আর আসি এবার লাস্ট ওয়ান দেখো ইনসুলিন completely different. It acts to a different receptor called TK receptor, tyrosine kinase receptor. Problem is, up to here it is very good. Below this level, that means below the belt. If you consider this membrane as a belt, then everything goes wrong below the belt. তোমাকে আমি একটা abusive word বললাম, you are using below the belt words, বলে না মানে কি unacceptable. So below the belt it is hostile. Below the belt is hostile. After this level it can go to any direction. There are at least 4 known pathways through which it can trigger even cancer. Insulin pathway. Insulin is a good hormone, it's not bad. But it promotes what? Positive metabolism or negative metabolism? Positive metabolism means anabolism. Cancer means what? Cells are more active, anabolic. So they are related. They are cross-linked. So below the belt, these pathways may crisscross with many of the cancer pathways. That's why when we go to block one pathway, other pathways might get affected. That is why it is very difficult to lodge a particular molecule against a particular cancer. Let us understand the same thing again. This is inactive protein, inhibitory action, and this is stimulatory action. Hormone binds. Clear? It comes to adenylyl. Adenylyl cyclase is bound to membrane by a 12 TM stretch of polypeptide. Fine? Clear? Inactive. L means ligand, R means receptor, alpha, beta and gamma. GTP binds, transient association, dissociates to form G active, then reassociate to regenerate the G protein. Correct? One example where cholera toxin acts. What is that? The step which is blocked by cholera toxin? Adenylation. এখানে যে জিডিপি বাউন্ড করছে, জিডিপি আসছে, এই বাইন্ডিংটাকে আটকে দেয়। Each diagram we are increasing one step ahead, more detailing. Cascades have certain advantages to include signal divergence, convergence and amplification. Means when required it can divert to other pathways, when required it can converge to same pathway, when required it can amplify. Suppose I am going to run in a marathon, so you must amplify the activity of the cell. Then a small amount of hormone will charge a lot of carbohydrate metabolism, lipid metabolism inside my muscle cells so that I can run the marathon. Amplification. Correct? Convergence, divergence. Divergence means we give for cell proliferation, it becomes cancerous, diverted. Convergence, we give medicine to kill a cancer cell, it kills a hair follicular cell. Convergence. We do it for something else, it happens something else. Inside the cell. It is so complicated and so difficult to understand. That is why. Just go through it. Adenylation bolam. Toxins, ADP ribosylation. This step is affected. Both of these toxins, ADP ribosylate, are gene residues in G protein and their activity. This reaction uses NAD and ADP. Each step. Bacterial toxins. Which one? Cholera toxins, pertussis toxin. লিখে রাখো পরীক্ষা আসে এটা পরীক্ষা আসে এটাই আসে শর্ট কোশ্চেনে আসে এমসিকিউতে আসে লং কোশ্চেনে আসে mechanism of action of bacterial cholera toxin, mechanism of action of pertussis toxin. খুব কমন they block ADP ribosylation এই লাইনটা মনে রাখবে কি মনে রাখবে they block ADP ribosylation of G protein. ADP ribosylation of G protein. ADP ribosylation of G protein. বলো একবার yes ভুলবে না পরীক্ষা আসবে দুটোর মধ্যে একটা এবার যদি কলেরা আসে পারতুসিস আসবে উত্তর একই কোশ্চেন আলাদা আসবে যার আগে পিছিয়ে যাই লেখো এই লাইনটা লিখো তাহলেই নম্বর পাবে আর আগে পিছিয়ে যতই কিছু লেখো না কেন এই লাইন না লিখলে নম্বর পাবে না ADP ribosylation it blocks ADP ribosylation ওই একই জিনিস করে ওইখানে দেখা যাচ্ছে কোথায় ব্লক করে দেখি জানো এই যে আরে cyclase stimulatory কে এফেক্ট করে ইনহিবিটর কে এফেক্ট করে এইবার এখানে আসি CDPK মানে is not CD of PK movie. CDPK means cyclic AMP dependent protein kinase. When I leave the slide here and go on talking, then you listen my words and look at the board, try to read along with it. তাই না কথায় বলে লেখা পড়া তাই না পড়াশোনা মানে কি পড়ো আর শোনো তাই না পড়ো শোনো লেখো তবেই কমপ্লিট সাইকেলটা পড়ো আর শোনো তাহলে সিডিপিকে একে কে অ্যাক্টিভেট করবে একে এইভাবে অ্যাক্টিভেট করা হয় সেটা কি R2C2 এই সিডিপিকে এই R2C2 যদি থাকে আর রেগুলেটরি সাবইউনিট রিলিজেস দি ক্যাটালিটিক সাবইউনিট এখানে দেখো এটা পড়ো these enzymes exist as কোন এনজাইম CDPK these enzymes exist as heterotetramer means tetramer means 4 subunits hetero means 2 subunits are not identical so heterotetramer means R1C1 R2C2 right R stands for regulatory subunit C stands for catalytic subunit so when this RC both are separated they are inactive when they are associated they are active ulto bole that the catalytic subunits when they are complexed they are inactive R2C2 is inactive cyclic AMP binds at the interface of R2C2 and then it dissociates so one cyclic AMP bound R1C1 another R2C2 they are two different subunits now they are active they can phosphorylate other effector proteins say this protein is getting phosphorylated to phosphorylated product in presence of ATP and enzyme is protein kinase A. Phosphodiesterase inhibit the production of phosphorylated protein product. এটা দরকার নেই, এই পয়েন্টটা মনে রাখবে, এইটা মনে রাখবে R2C2 এর ক্ষেত্রে CDPK। CDPK মানে কোন পাথওয়ে? গ্লুকাগন, এপিনেফ্রিন, নরএপিনেফ্রিন, এই পাথওয়ে, তিনটের পাথওয়েতে। দেখ, ligand receptor binding, activation of G protein, PLC means phospholipase C, activation of phospholipase C, phospholipase break down PIP2 to form IP3 and DAG, inositol triphosphate and diacylglycerol. Diacylglycerol being lipophilic, it travels through the membrane, within the membrane, and then see again it can regenerate. It is PIP2 can be regenerated by phosphorylation. This DAG goes to where? DAG goes to, এটা লিখে রাখ, ছবিটা আঁক বা ছবিটা তুলে রাখ, এটা আসে পরীক্ষায়। Activation of PIP2, activation of GI, activation of PLC, phospholipase C, breakdown of inositol triphosphate, phosphatidylate inositol triphosphate to IP3 and DAG. DAG travels through the membrane being lipophilic or hydrophobic, jai bolo, and then it stimulates protein kinase C mane related to phospholipase C, that's why C, nothing else. Now the problem. Phospholipase C is also activated by calcium. So this is a sign of convergence. Entry of calcium through calcium gated channel may also trigger the activation of phospholipase C, irrespective of presence of PLC or DAG. Phosphokinase C, PKC, protein kinase C কে অ্যাক্টিভেট করতে পারবে, IP3 ও করতে পারবে, DAG ও করতে পারবে, আবার ক্যালসিয়াম আয়নও করতে পারবে। So this is an example of convergence. ক্যালসিয়াম আয়ন দিয়ে বেরোচ্ছে, বললে তোমরা কি বললে, এন্ডোপ্লাজমিক রেটিকুলাম থেকে বেরোচ্ছে, তাই না? ট্রান্সভার্স রেটিকুলাম যেটাকে বলা, সেই ক্যালসিয়াম প্রোটিন কাইনেজ সি কে অ্যাক্টিভেট করছে, অন্যান্য কাজও করে, সেটা দেখবো পরে। So activation of protein kinase C then triggers the phosphorylation of effector protein. যে কোনো একটা প্রোটিন থাকতে পারে, যে কোনো একটা প্রোটিন, সেই প্রোটিনটা ট্রাইগ্লিসারাইড সিনথেসিস করতে পারে, সেই প্রোটিনটা কোলেস্টেরল ব্রেকডাউন করতে পারে, সেই প্রোটিনটা কোলেস্টেরল এস্টার ফর্ম করতে পারে। I don't know, we need not know unless this is the only pathway we have to study for 3 years. Okay, but it has got some destined effector molecules, that's all. This word is sufficient. What is the meaning of this? It is showing that protein kinase C may exist membrane bound or intracellular. Membrane bound or intracellular, it doesn't mind. Now these intracellular, don't think this is not membrane bound. It may be bound to some subcellular organelle's membrane. No kinase enzyme can be free, floating, dissolved in cytosol. Until they are free floating in the cytosol, they are inactive. Till they are floating, they are inactive. Until they are bound to some membrane. That membrane could be cell membrane, that membrane could be mitochondrial membrane, that membrane could be endoplasmic reticulum folds, trabecules, it may be Golgi bodies. Correct? Some or other membrane intervention must be there for activity of any kinase phosphatase enzyme. Wherever high energy compound is exchanged, those enzymes must be membrane bound, otherwise high energy compounds will get hydrolyzed will just dissipate energy, will not perform any effect. It is like cycling in one place. You will be tired, but you cannot move your feet. কি বলে এক্সারসাইজ সাইক্লিং, এটা তো আমি বলেছি আগেও। If ligand receptors binding, different ligand receptors binding stimulate similar type of G protein, it is convergence. If they diverge to different types of G protein activation, it is divergence. এবার এটা আছে insulin pathway. Insulin pathway. Insulin pathway is receptor is called tyrosine kinase receptor. Tyrosine protein kinase or tyrosine kinase receptor. It is actually a dimer. It is active when they are in close proximity. They are associated with another kinase protein called JAK kinase or JAK. The JAK kinase pathway, actually they cross phosphorylate, means this subunit will phosphorylate that subunit and this subunit will phosphorylate this subunit. It's called cross phosphorylation. So whenever an insulin hormone comes, these tyrosine kinase receptors, they comes close to each other and they hold the insulin and they cross phosphorylate each other, which triggers JAK kinase pathway. IR means intracellular receptors. These OH are phosphorylated. How? This JAK kinase, they phosphorylate. This subunit phosphorylate this one, this subunit phosphorylate this one. It's called cross phosphorylation. Now they can stimulate any of these pathways, say IPT DAC system, PIP2. এই জায়গাটা বুঝতে পারছো কি বলছি? When hormone comes near to the receptor, they comes in close association. And then when they come in close association, then phosphorylation takes place. This is a sign of phosphorylation. How? This segment, this subunit phosphorylate this one and this subunit phosphorylate this one. As a result, we get phosphorylated receptor. This phosphorylated receptor triggers what? PLC, phospholipase C, which triggers production of DAG and IP3. These DAG and IP3 stimulate protein kinase B pathway, activated protein kinase B phosphorylates the effector protein. So insulin tyrosine kinase acts to protein kinase B, glucagon acts to protein kinase A, the other pathways. আচ্ছা এটা আমি পড়াবো না ম্যাপ কাইনেস, এটা ক্যান্সার সময় পড়ানো হবে, ঠিক আছে? এটা আমি পড়াচ্ছি না, তোমরা এইটাই মনে রাখো, প্রোটিন কাইনেস বিটা মনে রাখো, খালি এইটুকু মনে রাখো। For this chapter, this is sufficient. Okay, up to protein kinase B. So tyrosine, insulin acts to tyrosine kinase receptor, then it activates phospholipase C. Then how it activates? Because it gets activated by cross phosphorylation. When they get activated, what they do? They activate phospholipase C. Phospholipase C produces DAG. DAG stimulates protein kinase B. Now, what is JAK-STAT pathway? Is there any regulatory step? Remember, in second messenger, there must be some regulatory point. What is that? The genus kinase or signal transduction activator of transcription systems, somewhat similar to TPK MAP kinase, tyrosine protein kinase and MAP kinase pathway. The JAK binds to a dimerase receptor. Which receptor? Dimerase receptor means konta? It can be tyrosine kinase receptor, it can be tyrosine kinase like receptor. Things are getting more and more complicated. Tyrosine kinase receptor was for insulin. Now there are many other factors which are generated by a cancer cell to trigger unusual effects. These molecules may also act through tyrosine kinase-like dimerized receptors. So through those receptors also, can you name one such hormone? IGF, insulin-like growth factor. It is not insulin, but it is insulin-like growth factor. So insulin acts to TPK receptor. Insulin EGF acts to TPK-like receptor. So what is the pathway for regulation of those pathway? That answer is JAK-STAT pathway, where a JAK kinase is associated with a STAT protein. Together they regulate the insulin-like growth factors pathway. Insulin is regulated by TPK MAP kinase pathway. Insulin is regulated by TPK MAP kinase pathway. Insulin-like growth factors are regulated by JAK-STAT pathway. Right? So here JAK binds to dimer receptor and phosphorylates them. Bind korlo, phosphorylate korlo. Right? Good. Cross phosphorylation. A korche. Now, the STAT then comes and binds with JAK. Janus kinase কাকে বলে জানো তো? Janus kinase নামটা কেন হয়েছে? In Greek mythology, there is a dog protecting the entrance of heaven. It has two heads. Who was killed by Hercules, I think, who was defeated by Hercules, something happened like that. I don't remember. So from there, Janus, because it is two-headed protein, hence the name came Janus. If the discoverer was Hindu, the name could have been different. We have any Hindu god with two heads? No, at least four, no? One, then four. Two are there? No. Any Hindu god with two heads is there? I'm asking you. No? Yes. No idea. Find out if there are two heads, so you can compare with that. So this Janus is associated with STAT, the activation of STAT, which translocates the nucleus where it acts as a transcription factor. Means what? The JAK remains associated with STAT. The moment two JAK molecule comes together and this JAK phosphorylate this JAK and this JAK phosphorylate this JAK. So now previously it was holding STAT. Now their structure changed. Now it is unable to hold STAT. So STAT falls apart from the JAK. It goes to directly nucleus where the DNA is standing and waiting for something which can trigger transcription process. এই লাইনটা মনে রাখলেই হবে। ক্যালসিয়াম তারই শেষ করছি একটু। ক্যালসিয়াম ক্যালমোডিয়াম দেখতে এরকম হয়, লাইক এ রাইট হ্যান্ড, দিস ওয়ান। এখানে ক্যালসিয়াম থাকে, এইটা একটা আলফা হেলিক্স, এটা একটা আলফা হেলিক্স, এরকম দেখতে লাগে। এইভাবে মাঝখানে ক্যালসিয়াম থাকে। এইগুলোকে সুপার সেকেন্ডারি স্ট্রাকচার বলে, যেগুলো বিভিন্ন প্রোটিনের মধ্যে পাওয়া যায়। ছবিটাও দেখা গেল, ছবি দরকার বাদে, দরকার নেই। আচ্ছা, এইটা নাইট্রিক অক্সাইডটা আমি কাউকে বলে দেবো পদে, এটা থাক নাকি? থাক, এটা এখানে থাক, নাইট্রিক অক্সাইড পরে পড়ানো যাবে। আপাতত আমরা কি শিখলাম? সাইক্লিক এমপি শিখলাম, সাইক্লিক জিএমপি শিখিনি না, একটু বাকি আছে। নাইট্রিক অক্সাইড আছে, সাইক্লিক জিএমপি। সাইক্লিক এমপির মধ্যে পরীক্ষা থাকবে, পরে পরে দেখা যাবে। up to cyclic AMP and different second messengers. Nitric oxide ইনসুলিনটা পরে দেখা যাবে। ইনসুলিনটা পড়ানো হয়েছে, নাইট্রিক অক্সাইড পরে দেখা যাবে। তাহলে এখান থেকে লং কোশ্চেন পাবে, মেকানিজম হরমোন অ্যাকশন, G14 মেকানিজম অফ অ্যাকশন, সাইক্লিক AMP রেগুলেশন, R2C2 CDPK অ্যাক্টিভেশন এবং ক্লাসিফাই হরমোনস। ওই প্রথম দিনে চারটা মনে রাখবে। ক্লাস 1, ক্লাস 2 হরমোন। ক্লাস 1 ইন্ট্রাসেলুলার রিসেপ্টর, ক্লাস 2 এক্সট্রাসেলুলার রিসেপ্টর। এইটা মনে রাখবে, এটা পরীক্ষা আসবে। একটা কোশ্চেন বলে দিলাম, ক্লাসিফাই হরমোন, এটা পরীক্ষায় দেবো, ঠিক আছে।